webinar series on advancing sustainable hydropower with a focus on biodiversity assessment and management. So we're very happy to have you with us today, and thank you for continuing to join us for this workshop of webinars in February. Today we're going to be hearing from two very esteemed researchers on macroinvertebrate sampling. So we're very happy to have them with us today to share their knowledge with us. I'm Leanne Alonso. I'm a biodiversity consultant with the International Finance Corporation. And I just want to say that this webinar series, our aim is to share information, particularly related to hydropower and its impacts on aquatic biodiversity, and methodologies and ways that we can assess and also manage and mitigate impacts on aquatic biodiversity. So we're glad to have a whole kind of range of different stakeholders joining us on this webinar series. This series is part of IFC's Hydro Advisory Program that's run by Kate Lazarus, and which is generously supported by the governments of Australia, Japan, and Norway. So we thank them for their continued support of the Hydro Advisory's work in Nepal. So before I get on, I just want to remind you that these workshops in February, these webinars are part of a workshop on the different components that make up the Trishuli assessment tool, which we presented on February 2nd, which is a standardized field method for aquatic biodiversity assessment and monitoring. So it's a methodology developed by a group of Nepali and international researchers to develop a more robust sampling method for aquatic biodiversity. So we're having this workshop on the different components, and we've talked about electrofishing and environmental DNA. And today we're talking about macroinvertebrate sampling, which is a very important component. And next week we will have fish identification and then data analysis, which are also very important parts of the tool. So we hope you have been joining us for all of the 6 webinars in this workshop. And just a reminder that if you attend all 6, you get a certificate and also consideration for in-person training that we plan to have on these topics. And I want to just remind you that we've added one more webinar to our overall webinar schedule about hatcheries. Rethinking Hatcheries, a review of the costs and benefits, will be presented by David Phillip and Julie Claassen on Tuesday, March 23rd. And they will also be presenting a webinar on telemetry and tracking of fish by radio telemetry and other methodologies on March 16th. So we're very excited to have this additional webinar because there was a lot of interest in hatcheries, it seemed, off the second webinar. So we wanted to have further discussion on this topic. So a few housekeeping rules for today. Remember that this is a 1.5 hour webinar, including the question and answer. You'll be automatically muted throughout the webinar. So we're also recording, so please be aware of that. Don't use the raise your hand for questions. If you have a question you'd like to ask the presenters, please put it in the chat, and I'll be compiling all the questions and asking them after the presentation. And if we have time, we can have some direct question and answer from the participants, I hope. If you have any issues administrative during the webinar, please send a private chat to Upasana Pradhan, who will then help you. And I'd like to thank Upasana for all her help in setting this up and getting all the flyers and really supporting this whole webinar series. So thank you, Upasana, for all your great work. We will be sharing the PowerPoint and the recording from this webinar and all the webinars with the participants afterwards. And you will receive an evaluation by email, and we hope that you can fill that out to help us out to make the next webinars even better. So the agenda for today, I just gave our welcoming opening remarks. We will have a presentation on macroinvertebrate sampling by Dr. Ramdevi Tachamul Shah from Kathmandu University and Dr. Deep Narayan Shah from Tribhuvan University. So we're very fortunate to have both of them with us today. I'm looking forward to their presentation. And then we'll have question and answer, after that. So I'm going to now introduce the speakers and ask them to please put up their slides. But before, I just want to say that, macroinvertebrates are extremely overlooked usually, but very important component of aquatic sampling. They form the basis of the food chain. They're very important in filtering water and play a lot of different ecological roles in the aquatic ecosystem. They're very important. They're also very good to be used as indicators of the quality of the water and of the habitat condition and status, which Ramdevi and Deep are going to share with us today. So I'm very excited about their presentation, and I thank them for joining us today, because they have developed a very robust and standardized method for Nepal that's widely used, and they've used it all across the country. So when we made the Trishuli assessment tool, we didn't need to come up with a new methodology for macroinvertebrate sampling, because they already had a very good sound methodology that we're just incorporating into the tool. So I thank them for already having that ready for us and having that thinking. So if you want to share your screen, please.
Yeah.
I will introduce you. So Dr. Ramdevi Tachamo Shah is a researcher at the Aquatic Ecology Center at Kathmandu University in Nepal. She holds a PhD in natural sciences from Johann Wolfgang Goethe University in Frankfurt, Germany. She works in the field of freshwater ecosystems, biodiversity, and climate change. She has over 15 years of research experience in Nepal, Bangladesh, Bhutan, India, China, Czech Republic, Austria, and Germany. She was a recipient of the TWAS Young Affiliateship Award 2020 Award for Central and South Asia. And she was awarded the Science and Technology Youth Award in 2016 by the National Academy of Science and Technology of the Government of Nepal. She's published more than 36 research articles in peer-reviewed journals, chapters, and 2 books. And then her esteemed colleague and husband, Dr. Deep Narayan Shah, is assistant professor in the Central Department of Environmental Science in Tribhuvan University in Nepal. He has a PhD in natural science, also from Goethe University in Germany, and over 15 years of experience as a researcher, manager, and consultant, largely in Nepal and China, but also in India and Germany. He has a master's degree in environmental science, focusing on water resource management and planning. He's particularly interested in this field of water resource management, but also freshwater ecology, aquatic biodiversity, and climate change research. He was awarded the Science and Technology Youth Award in 2017 from the National Academy of Science and Technology, Government of Nepal. He's published more than 36 research articles in peer-reviewed journals, several book chapters, and 5 books. So they are very world renowned experts on this macroinvertebrates and freshwater ecology, and I'm very happy to have them with us today. So I turn it over to you. Thank you.
Hello, good evening all, and thank you, Leanne, for your nice and very descriptive introductions. Now I would like to move on. So Leanne has already spoken about macroinvertebrates. These are the very tiny organisms, but can be seen with our naked eyes. And they have diverse kinds of ecological diverse kind of ecological roles in lentic as well as loctic ecosystems. So here today I would like to talk on different aspects of river and the biota, that is macroinvertebrates. So I'll be covering following subtitles, topics like river ecosystem and river health, macroinvertebrates, biological indicators, habitat assessment, how we'll take macroinvertebrate sample in rivers, both like vidable and non-vidable rivers. How do we identify these macroinvertebrates based on morphological characteristics? What are the measures we need to take in terms of safety, and what are the aspects we have to tech that is basically for quality control. And lastly, I will be talking about, or we'll be talking about data presentations, how we're gonna present the data that are collected in the field. So first and foremost, like river ecosystem, that is the river with water. It's a natural environment which consists or includes both living as well as non-living things, like various kinds of parameters like physical as well as chemical parameters, and how these biotic non-living parameters influence the biota living in the system. And we'll be basically talking about macroinvertebrates. So when we see in a river, it is a part of a landscape, and what we say is like rivers are biodiversity hotspot of landscape. And as a part of a landscape, whatever activities is happened in that landscape, it ultimately influenced the water quality and that govern the biotic communities. So there are different definitions of river health. The Webster dictionaries define health as a flourishing condition, well-being, vitality, or prosperity. An organism is healthy when it performs all its vital functions normally and properly, when it is able to recover from normal stresses. And in environment, when it is healthy, when the supply of goods and services required by both human and non-human residents is sustained. So that means if a river is healthy, that means it indicates the good conditions or good status. So health we normally use because this is a very easily perceived terminology and very much familiar to us. And these macroinvertebrates can be used as bioindicators because they live in water for most of their life stages, or some invertebrates actually spend their whole life in water. And these benthic macroinvertebrates are very diverse group of invertebrates that are retained on a zero 0.5 micrometer, that is 500 micrometer mesh size net, and can be easily seen with unaided eyes. And they chiefly comprise chiefly of insects, annelids, arachnids. You can also see a couple of photographs that I've kept on the right panel of the slides: crustaceans, clams, and gastropods. As I said, these invertebrates inhabit diverse habitats from very slow flowing water to very turbulent water to still water, and they adopt wide range of food depending upon their habitat preferences. That's why we can also categorize these communities based on functional feeding groups, which I will be talking later on. So in general, like benthic macroinvertebrates, benthic macroinvertebrates shows like they are very sensitive to different levels of disturbances. That's why around the world there are hundreds of methods that has been that are developed based on benthic macroinvertebrates. I've here kept a couple of examples. These are not limited to these photographs, but these are very wide range of organisms that I've kept here. The first panel is like mayfly. The first two figures are mayflies. Normally we identify these mayflies based on these three tails. And the down two are stoneflies, which are characterized by two tails. You can see distinctly in the photos. And the second panel are the examples of caddis flies. So these are they look like very much caterpillar, but they have jointed legs. And there are like water beetles, dragonflies, and damselflies, and true flies. like water bugs, dobson flies, shrimps, crab, snails, and like gastropods and annelids, uh, including like flatworms or leeches. So these are kinds of macroinvertebrates, and we need to know like which part of their life cycle we should look for assessing the river for assessing water bodies. So there are two different modes of life cycles they adopt. Most of the aquatic insects, they spend most of their life in water, but we need to identify like which stage is important, which can be applied in bioassessment. For example, this first one is an example of incomplete metamorphosis. One example I've kept here, like Plecoptera one, stoneflies one, second one on right panel is the dragonflies odonets one. So what we see is like there are only three different stages like egg, there are nymph, and there is adults. And these nymphs, like juveniles or mature nymphs, are very much similar to each other. So these are the complete metamorphosis. And we will be using this nymph stage of the invertebrates. And another one is complete metamorphosis, which consists of 4 different stages: egg, larva, pupa, and adult. And what we see is like larva, each of these stages is completely different than each other. Like larva is completely different than adult, which you can also see in this one. So in case of beetles, we use both like larva and adult for bioassessment. But in case of Trichoderma, we use only larval stage, because these adult stage, they live in, they have terrestrial mode of life. Macroinvertebrates, when we looked at, when we linked with river continuum concept, the biotic assembly or biotic communities in a river ecosystem is linked from upstream to downstream, because materials and other energy is transported from upstream to downstream. And what is interesting here is like at each stage, we see the functional feeding groups are completely different. Like in headwaters, we see the high compositions or like high proportions of shredders. But in case of lower middle river stem, we see like proportions of scrapper or grazers. And in downstream, where the river gets large and the river is slow, then we'll find large proportions of like biotic community is dominated by collector filterers, which feed on fine particulate matters. And here I've kept a couple of examples which belong to different functional groups. Like these are the first one is stoneflies one, which belong to shredders, like caddisfly, gamma rays, crustaceans. These all belong to shredders. And shredders means they feed on coarse organic particle matter, that is non-wood coarse particular organic matter, leaves, and twigs. That's why their proportion is normally high in the river stretches or like small rivers, which has high proportions of like the river is dependent on terrestrial food supply, like leaves, twigs from riparian vegetations. And the dominance of scrappers is high, basically in mid-river stretches. For example, these are the mayflies one, heptaginates. There are different families of caddisflies, like water pennies, water beetles, and collector gatherers. These are the organisms which collect coarse particular organic matter, and these are the major food for them. And the last one is collector filterers, which food is basically like fine organic particle matter or suspended particulate matter in rivers. So here just I wanted to give an examples, and here is one on right panel. We can see how each of these food materials is connected, and who is taking which kind of food. And basically, macroinvertebrates we use as a bioindicators, and basically it is also highly or very useful in assessing the impacts of hydropower dams. This is an example of Kaligandaki hydropower in Mirme. We can see like in upper stage, we see like water is impounded, but downstream of the dam, there is little to no water. And what we know is like if there is a dam, then the flow, material, everything becomes very different, because there is a disruptions of sediment transport from upstream to downstream. Flow regimes alter, habitats are destroyed, or there is alterations in habitat, which ultimately influence the composition and structures of benthic macroinvertebrates. And basically like there is changes in biotic assemblages, and which also can be seen or which also influence the feeding groups like upstream of the dam as well as downstream of the dam.
Hello again. Bioassessment, we use these organisms as in the biological assessment, because these biological endpoints provide the most integrated view of the river condition or river health, because these organisms, they live in water for a long time. They survive for weeks, months, and even a year. So these species, they integrate the condition for a longer period of time. This biological assessment process is poorly understood in different countries. However, this is increasing rapidly as a scientific foundation to support decisions regarding the protection of aquatic resources. Even in Nepal, the method is rapidly gaining its importance in the assessment. So when we talk about biological indicator, all organisms, particularly the macroinvertebrate species present in the river, may not be biological indicator. Only those organisms that respond to the environmental changes are considered as biological indicator. So when we define this biological indicator, we say that biological or animal species which, because of their ecological characteristics, react to a stressor or habitat degradation by clear and specific modification of their vital functions. So there are several group of organisms in the water bodies. For instance, there is macrophytes, there are fishes, there are macroinvertebrates, there are phytoplankton, zooplanktons. But why we consider these macroinvertebrates as more robust organisms for this assessment process? Because these species are cosmopolitan in nature and highly diverse. They are abundantly found in the river system, and due to their relatively large body size, it's easy to identify up to family level. And since these species are less mobile, it integrates the changes in environmental condition of a longer period of time. When we see fish, it's very mobile species. So when there's any disturbance, it quickly moves to other better habitat. So when we talk about the bioassessment, it's very important to know that This assessment considers high degree of standardization of the method. So we need to follow very strictly the standard procedure while doing biological assessment. For this macroinverse sampling, we need a number of consumable and non-consumable field gears, chemicals, and other materials. For instance, we need chest waders, hand gloves. We need life jacket if the water current is high. We need rope that needs to be tied in the waist. So we need sampling gears like kick nets, hand nets. We need bucket, tray, and many more gears that are listed here. In the beginning, we first collect the site information before sampling. And for this, there is a standard site protocol developed where we document the site description, the name of the stream. We put the site code. We document the latitude, longitude, altitude of the site investigated. We document the date and time and the name of the surveyor and investigator. Then we have weather conditions during the sampling. We document the stream characteristics, the catchment characteristics. We also document the stressor types, for example, solid wastes or effluents or activities and facilities or hydromorphological degradation, ecological degradation, personal hygiene and sanitation, and many other stressors types. We document hydromorphological parameters. Similarly, we also document several water quality parameters. And we also make a sketch of a river stretch indicating all these variables, particularly the stressor types, the river bank condition, the hydromorphological characteristics in the map carefully. So for this sampling, the site selection strategy is very important. For site selection, we consider both spatial and temporal scale. The site selected must be an appropriate river stretch that sufficiently represent the river. The number of samples should be enough. In general, for hydropower assessment, we consider 6 sampling sites above the dam in both main stem and the tributaries, 6 sampling sites in the dewater zone, both in the main stem and the tributaries, and 6 in the area of tailrace. So frequency of sampling over year is during base flow, pre-monsoon, and post-monsoon of the year. So generally we sample 3 times to document the annual condition of the particular river stretch river. At a site, when we identify for the assessment, we need to locate the different 20 soft sites. This means at each site, we take several samples and then we make a composite sample. So for this, we identify 20 soft sites considering the mineral habitat, the biotic habitat, flow type, the vegetation, and overlap environmental conditions at a site. So for this, we document the coverage of different microhabitat. So in this figure, you can see that in the left, there is a photo of a river showing the different substrate types. You can see there's a bedrock, covels, boulders, stones, sand, and on the right, there is a habitat assessment sheet. This habitat assessment sheet is used to identify those 20 soft sites. So this habitat estimation protocol can be filled first by identifying different types of mineral substrates, biotic substrates, and then estimating its coverage. And based on that, we identify the number of sampling units. So here you can see a river stretch where we can sample. And in this river stretch, we need to identify the different types of mineral substrate. So in this figure, you can see there is different microhabitats like mesolithal, akal, samal. So this mesolithal is the gravel, pebbles, boulders. So they have different types of mineral substrates that we need to first record them. After recording, we need to estimate their coverage. So in the second step, for example, mesolithal, the coverage is 55%. Akal, it's the 5%. Samal, the sand, it's 25%. The stones with algal vegetation covers 15%. And chilal, which is the fine material, that is less than 5%. And any microhabitat which is less than 5% is not considered for sampling, because here we consider at 5% interval. So after estimating the microhabitats, we assign the sampling units. So on the third column, you can see for mesolithal, now there is 11 sampling units. For akal, there is it's 5%. That means it's one sampling unit. And samal, its coverage is 25%. That means there is 5 sampling units. So you can see the red square in the river stretch indicating the sample units. For mesolithal, you can see the 11 sampling units are distributed throughout the selected river stretch. Similarly, for akal, that's the 1 point. It's shown in the river stretch. Similarly, there is samal, which is 25% coverage and has 5 sampling units. With this and stone algal, it's the 15% coverage that covers 3 sampling units. So in total, we have 20 sampling units. Now, once the sampling units are identified in a sampling stretch, then we start for sampling. So our sampling should be in such a way that it represents all the major habitats. So you can see on the right, the researcher is taking a sample. So for that, she has fixed her net against the current. So we always fix the net against the current. And then in front of the net frame, we disturb the substrate of the size 25 centimeter into 25 centimeter. So this means at each point, we take sample of 0.0625 meter square. And in total, when we take 20 samples, that means we are covering 1.25 meter square river bed surface in our sample. So for sampling, we roll up the substrate by foot or hand. And while moving the stones, we move up to the depth of 5 centimeter. We wash the cobbles, large woody debris by hand or using this soft brush. And once the sampling is taken from few sites, for example, for 2 to 4 soft sites, so we transfer the sample into the bucket because that may clog the net. To avoid clogging of the net, we keep on transferring into the bucket and take the sample. So this was all about the wadable rivers. So there are non-wadable rivers where we need to take the samples. For this, there are several methods that can be employed. For instance, there is artificial substrate. So in this method, we fill up the nylon bags with larger pores with stones of different sizes. and we place these bags in different points, in different strategic locations, considering the depth, the banks, and other macro habitat available in the river stretch. Once these bags are placed, we keep it for 2 weeks to allow the macroinvertebrates to colonize. And then we gently remove the bags from the river, put it in the large bucket. Slowly and gently we clean the stones, remove them, we wash the bag, remove it, and then we pass all the materials through a net having 500 micrometer mesh size, and we collect that for processing in the laboratory. The other method is to take the sample from the river bank. From the shoreline, we go inside into the river for certain stretch until half-meter water depth, and we take sample throughout the shore for 100-meter river stretch. So generally, we use these two strategies for sampling in non-viable rivers in Nepal. Once the sample is taken from all those 20 sub points, we put all those materials collected into a half-filled bucket, and then we process the sample. For processing, after transferring all the samples into the bucket with the half-filled water, we stir it and then pass only water with organic materials into the net. The mineral substrates such as sand, stones, gravels, or any wood parts remain in the bucket. We repeat the process for a few times until all the organisms are passed into the net. So at the end, we check the remaining materials in the bucket carefully, and if there is no any organism remained in the bucket, we discard the material from the bucket. And then we transfer the materials that were retained in the net into the white tray. And then we pick the organisms into the petri dish. We check for any protected species, any red lit species. If there are any, we document them and put the organisms back into the river. Also, we look for any organisms which needs special care. So we pick them and preserve them carefully in separate vials. So all the samples collected, they are carefully placed them into the vials, bottles, buckets, and labeled carefully. We label them with sampling code. We add the site name, the sampling date, and total number of containers. Sometimes we have more than one containers or the bottles for sample. So we use, for example, if there are 4 bottles, we number it as 1/4, 2/4, 3/4, and 4/4. This indicate that there are 4 sample boxes for that particular site, so that it becomes easy to sort and identify and put them together in the lab. And only properly labeled samples are then fixed and preserved with standard preservatives. So standard preservatives are ethyl alcohol and formaldehyde. So ethyl alcohol, we use 99.9% conc ethyl alcohol, and for formaldehyde, we use 4% formaldehyde. So if we normally recommend all the samples to be stored, preserved in ethyl alcohol. If the sample has to be fixed, the formalin is used. And if formalin is used, it needs to be stored for 2 weeks, and then further treatment is required. That is, the formaldehyde needs to be washed, the sample needs to be sorted and transferred into the preservative, that is ethyl alcohol, for longer time period. So once the sample is brought into the laboratory, then the samples are processed. For processing, first the sorting of specimens is done. All the macroinvertebrates are sorted from the sample. They are separated at the order level and then carefully identified taxonomically up to family level, genus level, or species level as required. There are several taxonomic identification keys available now. There are several research articles with different orders with identification information. There are books for non-insects, insects, which can be used for identification of organisms. There are several advantages of multi-habitat sampling because this is the quantitative method. Therefore, the number of species recorded for site can directly be employed to statistical techniques. This is a representative copy of a site because, as I mentioned earlier, we use 25 centimeter by 25 centimeter area for each sample point to collect the sample. And when we take 20 sampling units, that means we are covering 1.25 meter square, which is then converted into per meter square. So that gives the representation of that particular site. It provides abundance of each group in addition to composition. So it not only provides the richness, but also the abundance of each organisms at a site that can be further analyzed in different ways for different purposes as required. For example, we can also use the data for functional feeding groups analysis. So there are some cons for this multi-habitat sampling. This habitat does not include the sample from underrepresented habitat, which is less than 5%. This is why this method does not mean for collecting all the species available at that site. So if we want to get the entire species list of that particular site, we use qualitative method where we can sample until we get, we keep on getting new species at that site. So to get the complete taxa list, we use qualitative sampling approach. The figure on the right shows the net that we use for qualitative sampling at a site. We need to take care of these quality control measures, that all the samples collected should be properly placed into the sample container. The labelings should be done both inside and outside the box with the same information so that it's more secure. If one is lost, the other label can be used for identifying the site. All the gears, particularly the nets, needs to be cleaned so that the same net or gear used in other site may not duplicate the species collected from one site to other site. So it's always important to examine again and again all the gears used for sampling in other site. The other important is, as we are working in the river may have different level of water, and that may be dangerous sometime. So we need to take care of the safety measures. For instance, we should always put wading trousers. We should use the life jacket. We should also tie the rope in our waist, and someone on the bank should take care of it. We should have our first aid kit with us in the field. So there are several safety measures that needs to be considered while being in the field.
So when we, based on different kinds of disturbance, different kinds of perturbations in a site, and the rivers is normally categorized, classified into 5 different classes, like good, high, good, moderate, poor, bad. High and good are the desirable status, and when it turns to like moderate, then it needs restorations, and basically poor and bad, they needs immediate attention for restorations. There are different ways of analysis, basically like the EPT, which are considered as EPT means like mayflies, stoneflies, caddisflies, which are considered as some of the sensitive groups in benthic macroinvertebrates. And this EPT index is widely used in different countries for assessing the water bodies, basically like streams and rivers. And that EPT index is calculated by counting the number of Ephemeroptera, Plecoptera, and Trichoptera present in a site. And there is classifications we can see here, how it is rated into different water quality, like excellent, good, and fair, poor. So based on number of EPT taxa present on the site, we can classify, we can rate the site into different classes. And there are other candidate metrics which we have proposed actually based on different literatures. And these are categorized into different metric types. We can look at richness measures. In case of richness measures, total number of taxa richness in a site could be one. EPT richness, which is similar to like EPT index, is the another candidate metrics. And there is also like COH, that means number of present Coleoptera, Odonata, Hemiptera taxa, Diptera richness, worms and leeches richness. And the quantitative measures would be like Shannon-Wiener diversity index, evenness index, Simpson diversity index. Another measures is composition measures. These are the relative proportions of each of these groups, like percent of EPT richness, which can be both in terms of abundance and richness. If Meroptera, mayfly richness, Trichoptera richness, COH richness, and dominant tree dominant taxa. So depending on different types of river stretches, these proportions actually changes, or depending upon the disturbances, perturbations in a river, these matrices or the value changes. And the value is, some of these sensitive taxa is significantly lower in impacted sites compared to the reference sites. But there are some of the measures like number of percent of Chironomidae individuals, like percent of mollusks individuals, which could be increased in impacted sites compared to reference sites. Another measures is sensitive measures. And sensitive measures, we looked at organisms which are sensitive to different levels of disturbance. We have categorized here three different levels. One is sensitive taxa, which has score, tolerance score equal or greater than 7. Another one is moderate taxa richness, which has taxa tolerance score between 4 and 6. And another one is tolerant taxa richness, which tolerance score, or which has tolerance scores of 1 to 3. So this tolerant taxa means these are highly tolerant to disturbances. Some of the organisms may survive in even in very low oxygen, like below 4 milligram per liter of dissolved oxygen, and in organisms, sensitive organisms, sorry, sensitive organisms means like which are highly sensitive. They can be found only in clean water bodies with high dissolved oxygen, like dissolved oxygen of 6, 7. So these are the metrics we can employ in assessment. And there's another method like biotic index method, which I'll be also showing how we can calculate this biotic index, which is basically calculating tolerance score of each of the taxa present in a site divided by total number of score taxa in a site. Another one is functional feeding groups. I will also show an examples for Trishuli River with impacted and non-impacted sites, how the proportion actually change. So there is shredders richness, scrapper richness, collector gather richness, filterer, their abundances can be also used in assessment. And in case of higher level one, we can use the biomass or dry biomass of all individuals present in a site. So this is an example I've calculated for for a reference site. This is Upper Chilime, first one, and on right one, we can see the river stretch, that is the dewater zone one. And what we distinctly see is like the proportion shredders actually dramatically changed from reference sites to the dewater zone, and that is basically because of the reduced flow, and it has discontinuity. There is discontinuity between riparian zone and main water channels. And what we see high proportion is like collector filters that are basically dependent on fine organic particulate matter with matters. And another one is like APT index one. I've calculated for different site APT index for Upper Chilime, and what we see is like basically in a reference site, we see good to fair water bodies like Melung it's one of the reference site we've considered in Trishuli hydropower assessment, and Tadi is another reference sites we have considered, which has maintained higher number of EPT taxa. And these EPT taxa, since most of these organisms are sensitive to flow regimes and other water quality parameter, the values get lower in impacted sites, basically like below Trishuli Dam 8, or like tends to be poor, and also like the water zone, which is 12, fair one. So this is the determination of river quality class, which I've calculated for a site. And this is normally done based on the number of taxa present or type of taxa present in a site. And on right column, we can see a taxa sensitive score. This YTD 4 means these are moderately tolerant to perturbations or disturbance, and like Perlidi, this is sensitive to disturbances. And these are basically the scores are given basically based on the auto-ecological requirement of requirement. 8 or tends to be 10 means highly sensitive, 10 is highly sensitive to disturbances, and 10 mean highly tolerant to disturbances. And how we can calculate is biotic index equal to, this is summations TSSI, total number of sensitive score divided by total number of score taxa. For example, here we have altogether 12 taxa in a site, and we do not have score for one taxa, that is Synlistidae. This is damselfly. So if we do not have sensitive score for taxa, we do not count in this in the index. So the sum of each of these values gives us 84. That is 84, and divided means though we have 12 taxa present in a site, we have considered only 11 because we do not have we do not have sensitive taxa score for one side. So 84 divided by 11 means 7.6. And this final value, calculated value, is tallied with a river quality class transformations one. And based on that, we will get 5 different river quality class. So here 7.6 means it gains one, that means high status one. So we give this river quality class translate into different bandings, different color band. And why we do it? Because it is easily understandable transformations of complex scientific informations. Instead of telling like number or sensitive or tolerance taxa to non-technician people, if we show with the color bands, it somehow helps to capture the informations that we are trying to give. So these are the couple of maps that we have prepared for different water sets of Western Nepal, like water sets of Karnali River Basin. This one is for lower Karnali water set, Puligat water set. This we prepared for the USAID PANI program. And this is for Chimbruk water set. So that means like blue means high quality, green means good, still good quality, yellow means the river stretches are moderately polluted and moderately disturbed. And that basically this is the site downstream of the Chimbruk hydropower dam. The all yellow zones is the like upstream of here is the Chimbruk dam. The yellow is the impounded areas, and downstream is the dewater zone. And this is for Tuligat watershed. So all these informations are available in our recently published handbook, Rivers Handbook, and you can get the handbooks at Aquatic Ecology Center, Kathmandu universities.
Since we are using this organism as a tool to monitor our river systems, so every time the expert doesn't need to visit the site to sample, we can train the local people, also called as citizen scientists. As the method is very simple, they can collect the sample, they can identify with the help of pictorial key and document the status. So here we have conducted the training in the Western Nepal to 25 citizen scientists who were trained in the river for taking the sample using these standard methods, sorting them, collecting the specimens, identifying them. You can see the pictorial key in their hands. We have prepared the manual for the citizen scientists. So based on the manual, they follow the standard procedure and collect the sample. So this helps to take large number of sample in a particular time, and we can monitor the river stretches in the entire basin at a time. Thank you so much.
Great, thank you very much, Ramdevi and Deep. Very interesting, and I really am interested in macroinvertebrates. I was telling you that when I first saw a dragonfly nymph like this one in the picture, that's a mayfly, right? I thought it was like a space alien. They're so weird looking, but they're very diverse and very interesting and play a lot of important roles. So I had to get over that initial shock. So, and I think other people too, well, once they see more of them, they'll really appreciate them. So thank you, that was really interesting. And I'm going to ask a few questions now. So You can unshare your screen if you like. Yeah, good, thanks. And we'll just have it on. Okay, I'll start with the ones that I've already, questions I've already kind of accumulated. There are some questions about the method, some about taxonomy, and some about like the use, the analysis and the use of the method. So I'll group them in that kind of way. So I'll start with one that's maybe kind of, I'm not sure that it's a bit challenging, but there's several questions about what do, if you see changes in the macroinvertebrates, or what do the macroinvertebrates, can they tell you about fish? Can they tell you about the size of the fish populations or changes in the fish population? I know they're the food. So if you see which invertebrates, if you might see changes in, would that affect fish more? So can you tell us if they tell you much about fish? Oh, you're on mute, sorry. You're under mute, yeah.
Sorry. The presence of a particular macroinvertebrate doesn't indicate the presence of particular fish species, but in general, the abundance, the biomass of macroinvertebrates indicate the presence of fish, because these all organism groups, they're dependent on each other in the food chain, in the food web. So as they are the part of food, as you mentioned, so if there are enough food, because these organisms, the macroinvertebrates, they're dependent on the producers, the algae, or on the leaf litters, at the energy level. So again, they are eaten by the fish. So if there are a good number of organisms, the macroinvertebrates, the abundance, the biomass is high, you can predict that the fishes are also present there. And in the same time, if the river system is healthy, the presence of macroinvertebrates indicate this. So, and if the river system is healthy, so we can predict that there is fish species, if the habitat is enough.
Very good. Yes, I imagine there's a good, close relationship between that. Do we know much about which invertebrates fish eat, or certain fish are eating? Do we know much about diets
For
of
example,
macroinvertebrates?
yeah, in Nepal, if you go to the rural areas, you see that the fishermen, they collect the Heptagonidae, the larger ones, and use them as a bait to fish it. So that clearly indicates that the fish, they eat these species.
So we could maybe get some inference in terms of what fish could be there, in terms of if they eat those kind of invertebrates.
Yeah.
Okay, thank you. I'm going to move over to a little bit about the method. So what about water temperature? How does that affect macroinvertebrates and your sampling?
Okay, so these benthic macroinvertebrates are also widely used to see the impact of water temperature. Because some of the benthic macroinvertebrates are highly sensitive. That means not only to the dissolved oxygen, but also water temperatures. So there are organisms which live in cold, cool water. These are the sensitive taxas, and the same taxa we do not find, like the species that we find in mountain regions, we do not find the same taxa in lowland regions. So that is basically due to another is due to temperature difference. That's why in one of my study, I have looked at how we can consider or how we can utilize these properties to detect the impact of climate change on river ecosystem. So I've identified 33 different taxa, benthic macroinvertebrate taxa, that are particularly sensitive to temperatures. So that means like these are climatically sensitive. That's why they can also employed to detect the effects of climate change or like warming trend in water bodies. And basically, in case of like cold water bodies, we'll find a high abundance of EPT, Ephemeroptera, Plecoptera, and Trichoptera. But in case of warmer temperature, we see like water beetles, water bugs, or dipterans. So we distinctly see the compositions and different compositions in cold water and warmer water bodies.
Great, thank you. Yeah, the sensitivity to those things make them good indicators, since if it changes, you can see it right away in the macroinvertebrates, right? There are other questions about climate change, but I'll come back to that. There was a question about how to sample in deep and high currents. And then Bill was asking, have you tried hand-thrown dredges for sampling the deeper water?
Yeah, this is challenging as well, I would say. It's easy to sample in the wadable rivers where we can go at different macro habitats. We can cross the river and take the sample wherever we want to take. But that doesn't happen in the non-wadable rivers. We can't reach all the sites. Internationally, there are several methods used. The big cranes, the wedges are used to collect the samples. But here, particularly in Nepal, we use these two techniques. One is the artificial substrate, as I mentioned before, in the nylon bigger mesh size bag, we fill up with stones, sands, and we place them in different strategic locations. And then we keep it for a certain time, at least for 2 weeks, where the organisms it colonizes, and then we collect them. The other method is we take sample from the source, and that is we go into the river until half meter to 50 to 60 centimeter water depth, not more than that. And sometimes maybe we enter 5, 10 meters, 20 meters until we get more depth than half meter, or sometimes we get such depth immediately at the edge in the shoreline. So it depends on the river morphology. So in this case, we identify different microhabitats along the shoreline until where we can reach.
Great, thank you. Do I still have my thing? Okay, good, thank you. Yeah, I'm sure it's challenging to do the high current. And what time of year, I'm not sure, did you mention what time of year is best for the sampling? Is it similar to the other taxonomic groups?
Except monsoon, because in the monsoon, the water level gets high because the Nepalese rivers are monsoon influenced by monsoon. So the river gets high, and the main river channel, we can't reach to the main river channel. The water floods, the flood plain increases. So at the edge, we get nothing because the organisms are flooded, the water is turbid, the velocity is high, depth is high. So we avoid the monsoon, and then we take in other 3 seasons. For example, in the pre-monsoon, in the post-monsoon, and in the base flow time.
Good, yeah, thank you. Yeah,
I just wanted to add on to the questions with deep and high currents one. So basically in bioassessment, we compare with respect to reference sites, like impact sites versus reference sites. So basically, if the river stretch is, if we are working in deep and high current water rivers, then we need to actually compare the impacted sites. It needs to be also compared with the similar one, like the high and large one. So we cannot simply compare with the small tributaries or wideable river stretches where we can rigorously take samples. That makes like lots of effort, and we get high abundance of organisms. And basically in deep and high currents one, since we are using different techniques, it needs to be comparable. It needs to be compared with the similar river types and river size.
That's a good point with the monitoring. There was a question about reservoirs in terms of, do you see differences in, is it hard to sample reservoirs? And also, do you see differences in the macroinvertebrate composition?
Yeah, of course, we see different composition in the reservoirs than the running water stretch. This is because the oxygen level is different, the temperature is different, the nutrient level is different. So the organism groups changes, obviously changes, and that indicates the difference between the free flowing and the standing water of the reservoir.
Thank you. Okay, about some taxonomy questions. So how well is the taxonomy done, well known for invertebrates in Nepal and the Himalayan area? And are there any online resources for identifying? Also, could you explain what level? Because I know there's different levels that you need to identify them to, like order, family, and to what level do you need to go?
If we see the situation regarding the taxonomic identification and available materials, it's at the infancy, I would say. It's still growing stage. We don't have much literature in the Himalayan region, but since last decade, I would say the number of literatures are growing, and we are getting more and more publications in this subject. So now we have most of the keys available up to family level. So for family level taxonomic identification, I think it's easy to carry on. But at genus and species level, it's still not that easy, because we need to compare, we need to establish first. We need to compare the larvae with the adult and see the characteristics of these particular species. But in the same time, we can take help of the existing keys from China, from Malaysia, from North America, though the species do not, though the same species do not occur in different parts, but that helps in the identification.
Okay, good, thank you. Yeah, I mean, at the higher levels, you can use those order, family, and I know a lot of your, some of the indices only require that kind of level, right? Rather than species level.
Actually, yes, we have tried in a couple of research, like how order level is sensitive to different kinds of disturbances. And basically, if there is a like distinct type of disturbance, like organic pollutions one, we can see clear distinction at order level. But basically, like in case of hydropower or like flow regimes, that level doesn't work actually. So fine resolutions is essential to get precise result.
Can you use, do you use morphospecies a lot? Just giving your own, just sorting them into different species, but without a real name?
That is possible, but the problem with that one is like, because it's individuals see differently for each species. So maybe some people may not recognize differences between two different taxonomically different organisms. So that means like somehow it will disturb the data resolutions, or let's say like data itself.
Okay, thank you. Among your indices that you showed various metrics. Which ones do you use most often for the studies that have seemed to give some give results that are useful?
Yeah, so though there are different kinds of like composition measures, richness measures, or functional feeding groups measures, not all are sensitive to different kinds of disturbances. For example, taxa richness may be useful when there is organic pollutions. But in case of like hydropower or like flow alterations one, habitat alterations one, maybe EPT abundance work. So EPT abundance normally like trachyptera abundance work, which we saw from the Western regions. And another one is like functional feeding group. So the functional feeding group matrix works perfectly in different kinds of disturbances, including like morphological disturbances.
And how do you use these different, how do you use the results then and these different analyses? What in order to convey the message to the like a hydropower company, how can they use this information on macroinvertebrates and your river classification system? What really should they be getting out of the kind of results coming out of your studies?
We can use different methods. For example, first, as shown during the presentation, the river quality maps. That maps are created based on the biotic index. So based on the biotic index or sensitive taxa score index values, we can classify the river sections into 5 classes and plot them using different color codes in the drainage map. And that gives very representative of the river basin, where you can clearly see which river section has which class. And if it degrades more, for example, it converts from blue to green, green to yellow, yellow to orange, orange to red, then we can prioritize our restoration in that particular stretch. If you go into more in depth, we can look for the EPT index and see how this index value increases or decreases. Similarly, we have composition measures, or we have the sensitive taxa measures, or even the functional feeding index groups. So we use different index values to compare from one sites to other sites and from one time period to other time period to see the variance of that particular site.
Right. So since you're monitoring and doing this over again and again over time, the changes in those classifications of the river should give the hydropower projects an idea of how the river is changing with their operations. Yeah. And I like the colors and the maps, because that is a good way to show to people who aren't scientifically inclined and to make the information clear right away. There are a couple questions. We'll have a couple more questions and we'll have to wrap up. Couple questions about climate change, and I know you're both interested in that. And how can your macroinvertebrate data, can it be an indicator of climatic events, and how is it? Have you been seeing any changes over time in the work you've been doing in Nepal?
Here, as Ramdevi mentioned it before, that we had conducted research in the central Nepal. We have collected samples from 1,000 meter to 4,500 meter altitude above the sea level, and from different habitats from the natural river conditions. So what we found that the rivers, the altitude between 2,900 to 3,400, they are very sensitive to the climate change. And based on this concept, the sensitive taxas, sensitive to climate change, that means sensitive to temperature, was identified. And in total, 32 species are identified basically for monitoring the effect of climate change. So there is existing method. The species are identified for using it for monitoring purpose. So these identified species can be used for monitoring purpose.
Yeah, addition to that, we have also carried out a couple of research where we looked at how future temperature or future climate change could impact the sensitive sensitive invertebrates. So some of like couple of examples I could take here is like relict Himalayan dragonfly, which is also listed as IUCN threatened, near threatened taxon. So we actually simulated across Hindu Kush Himalayan regions, and what we found is like there is restricted areas with where it is today, and habitat is shrink quite dramatically in near future. So that is in terms of like climate warming. So climate warming will impact definitely their habitats. So climate change, like to other organisms, in case of invertebrates also, like the climate change will impact to the cold adapted species rather than warm adapted species. So the taxa that are that inhabit in high mountain, like in mountain streams, will be more vulnerable in terms of climate change compared to the taxa that are that are inhabited to the lower river stretches or like in warm water bodies. These are very sensitive, also vulnerable also because they have limited mobility. So they cannot just move upstream or downstream with increasing temperature. And basically, like the organisms which are sessile, they will be more vulnerable to climate change, like snails and other like annelids.
Right. Okay. Thank you. Yeah. Definitely isn't they could be it's important to monitor them as we have this changes in our climate. So two more questions. So if you're using it macroinvertebrates to monitor like restoration or a change in river, preferably going better, do these macroinvertebrates naturally kind of get restored as you see changes, or do you ever like have to introduce any of these kind of taxa that were gone? And then are there any, have you seen any positive impacts of hydropower on macroinvertebrates? Are there any places or taxa that are seem to do better?
It's really very interesting question. When we talk about the restoration and think of restoring the habitat, it's always the species pool zone nearby. So this is why when we deteriorate or disturb the main channel, the tributaries may act as the species pool areas. So where organisms, they survive in these hard times. And when the main stretch gets restored, the species from the nearby species pool zones, they get into the main channel. So this is why we need to always conserve the tributaries in case of also the hydropower in all the sections above dam, in the dewater zone, and in the trailless areas. We need to conserve the tributaries so that the species occurring in that areas are they remain in those tributaries, and that serve as the species pool zone when the other part is restored.
Okay, yeah, definitely. And do you ever see, are there any taxa or habitats that are positively impacted by the hydropower?
Yeah. for example, the reservoir, it acts the different habitat. That's it's the Atlantic zone. So in that Atlantic zone, we gradually get these species, particularly that suit into these standing water bodies. For instance, the odonates, and they serve as the suitable habitat for many species that were not present in the
Ah,
straits.
right.
So that can act as a refuge areas for such species, particularly in the context of climate change.
Gotcha. That makes sense. Good. Okay, well, I think we're out of our time now. And so I'd really like to thank you both, Deep and Ramdevi, for a very interesting presentation and for inspiring everyone to consider macroinvertebrates and to come to love them, I hope, as much as you do. But they are very useful and super important in the aquatic ecosystem, so we should not overlook them. And we must continue to consider them in these assessments and in the impacts of all the things we're doing to these rivers. So thank you very much for your presentation today.
Thank you
I
so
just
much
want to-
for
Thank you
giving
so
the
much.
opportunity
Yeah.
to share
Great.
our.
I just want to remind everyone that we have 2 more webinars in this workshop next week, but next week is on Wednesday, not Tuesday, because of the Nepal holiday on Tuesday. So on Wednesday is on Himalayan fish identification, which is going to be very interesting about the resources, but also the current status of taxonomy, and how we can start to work on that. And then on Thursday, we'll be on data analysis for long-term monitoring. And then remember, in March, we have two other webinars as we continue the series. So thank you, everybody, for joining. Really appreciate your continued participation. And we look forward to seeing you all next week.
Yeah.
Thank you.
Thank you.
Thank
Thank
you.
you.
very much.
Bye bye.
Bye-bye.
Bye.